interpretation of the cell cycle of an animal cell. First is interphase. Here you can see the blank cell. Now the cell has the nucleus with condensed chromosomes and the cell with centrosomes.
Next is prophase. The chromosomes are now uncondensed and each pair of sister chromatids are joined together. Now the centrosomes appear with their spindle fibers. As you can see, the nuclear envelope has disappeared and we are just left with the chromosomes.
After comes the prometaphase, our centrosomes with the spindle fibers, but now they are spreading throughout the cell. And now the chromosomes are at the end of each spindle fiber. As you can see, the chromosomes have attached themselves to the end of the spindle fibers and are spread throughout the cell, ready for metaphase.
After that preparation comes metaphase. The centrosomes and the spindle fibers are now reaching towards the middle of the cell. Finally, the chromosomes at the end of each spindle fiber line up in the middle of the cell. Each spindle fiber is attached to the centromere, which is holding the chromatids together for now.
Next is anaphase. Now the cell with the spindle fibers and centrosomes, again, but now pulled back a little bit. After that, the chromatids are separated and pulled to the poles of the cell by the spindle fibers. As you can see, the chromosomes have separated and are now just chromatids.
After that is telophase. Again, we have the cell, but this time stretched a bit and pinched in the middle. Now the nuclear envelope has chromatids inside. Last is the centrosomes, this time without the spindle fibers. As the cell pinched, the nuclear envelope reformed and the spindle fibers retract as the chromatids were enveloped by the nucleus.
Our final stage is cytokinesis. Now there are two cells completely separated. Now the nucleus with the condensed chromosomes reappear. And finally, the centrosomes pair. As you can see, the cell has finished the process and looks similar to what it looked like in the start. This is because the same process is going to happen again and again with these two new cells. Misconceptions. Misconception number 1. In early development of organisms, cells that result from cell division do not grow and divide again. In the early stages of organisms' development, cells do divide again after they divide once. If they did not continue to divide, an organism would not be able to grow or be able to heal wounds.
Misconception number 2. Organisms grow by cell division, but the cells themselves do not increase in size or mass. Organisms grow by cell division, but the cells also increase in size and mass. If the cells did not grow after dividing, there would eventually not be enough material left to divide again, and the organism would not be able to grow and heal wounds.
Misconception number 3. In early development of organisms, cells grow in size, but the number of cells remains constant. As we discussed in the last misconception, cells do grow in size, but they also do grow in number. If cells only grew in size, the membrane of the cell would not be able to keep up with the organelles. As the cells grow, the organelles grow at a faster rate than the membrane, and the membrane can't transport enough food and oxygen.
Misconception number 4. In early development of an organism, the organism grows in size and mass without cell division or cell growth. This is incorrect because if the cells did not divide or grow, how else would organisms grow? As we know, matter cannot be created or destroyed, so the development of organisms is not an exception. Cells cannot just randomly come out of nowhere and join an organism's body so they can grow.
Misconception number 5. Cytokinesis is part of the process of mitosis. This is wrong because cytokinesis is its own step in cell division along with interphase. Mitosis is the division of the nucleus, which does not happen in cytokinesis.